Hi everyone, my name is Carly Boyd. I'm from the Zeller lab at SDSU, and today I'm going to be talking about comparative genomics, the new cool tool in scientific research. So most people know about DNA, and we know that it is composed of nucleotides encoded by letters A, C, T, and G. And these can code for either a protein or a certain function. But actually, the individual genes aren't the whole story. They're just a paragraph in the book called the genome. Every living organism has a genome that has all of their DNA in it, such as a human, a mouse, a fruit fly, and a sea squirt, which is what we study in my lab. You can look at all of these genomes and compare them for their percent identity to each other, which is the basis of comparative genomics. You can do this computationally, and right here I have an example of a graph. It would be called a vista plot, and on the bottom you would have like the entire genome aligned together, both whatever you're comparing, and then you have the percent similarity, and then these peaks and these valleys down here represent areas with high similarity or low similarity. So basically, what we try to do is, if you know that in a human you have like sensory neuron development or something like that, and you know that a mouse does as well, most of the time that's what we do our experiments on, is a mouse. But what if we could also do an experiment on something like a sea squirt and use that in the lab? So scientists first start out by looking at the genomes. So you have the genome of the whole human that you're looking at. And again, you're doing this computationally. So this circle represents the entire genome of the human. And then you can take the entire genome of a sea squirt and you compare them to find out what parts of their genome align, which is represented right here. And you're trying to find out what cells or what genes and part of the genome codes for like sensory neuron development. So then, after you do that, you can take an organism that you know does not have neuron development, like a plant, and you can cross out all of this and whatever aligns to this, because we know that that's obviously not going to code for neuron development, because that's not what a plant has. So then you're left with this right here, this chunk of the genome that are similar between the human and the sea squirt and do not include anything that wouldn't be for sensory neuron development or neural development. So you go from having thousands upon thousands of genes that you're trying to look at to basically maybe 10 to 100. And then from there, you know that you can perform experiments in the lab on a sea squirt with these genes and see what they do knockout experiments and that type of thing, which can save scientists a lot of time and a lot of money. And that is why comparative genomics is the new cool tool in scientific research. Thank you for your time. Please make sure to hit that like button, subscribe, and I'll see you soon.
Thanks for watching another presentation here at the science class. If you want to know more about the research that you just heard about, check the video description below for a link to that lab's website. And if you want to see more presentations like this in the future, you can subscribe to us by clicking this button here. Thanks very much.